Hey everyone, welcome back. We are here on slide 10 of chapter 19. Slide 10, chapter 19 of senses. We've previously discussed different receptor types based on structure. Here we're talking about specifically about the senses that we have in our ear. It's not just hearing. Hearing is one of them, but it's also our vestibular sense. Vestibular sense is our sense of movement and balance, head movement and balance. So to lead into that, let's talk about structurally what our ear is like and what outer ear is like leading to our inner ear. Our ear isn't just the pinna. It's everything from the pinna and deeper as you go in the ear canal. So we can divide up the ear into three different parts. The first part here is the outer ear. The outer ear, external ear shown here, includes all of this. It includes your auricle or pinna. This is shaped round like this so that it can funnel in sound from different directions. If you scrunch up your ear like this, you can still hear. It's just gonna be harder to locate in 3 dimensions where that sound is coming from. And this auricle, remember, is made of elastic cartilage so that if you brush your head up on something in tight spaces, no problem, it'll just bend and adapt to whatever it's brushing against. This space right here, this is our external acoustic meatus, the external auditory canal leading in deeper into your ear. In this external acoustic meatus, you have skin, and this skin is lined with a special gland. It is lined with ceruminous glands. Ceruminous glands produce cerumen. Cerumen is a fancy way to say earwax. And remember that this gland is an apocrine gland. It secretes by shedding off the apical side of the cells, much like mammary glands or apocrine sweat glands, apocrine pseudoryphals. Earwax, why do we produce it? It is antimicrobial. It catches any particles that might get in deeper. It is a problem if you get too much of a buildup, but that's the goal of it, to help catch things and prevent growth and prevent infection. The deepest part of the outer ear is the tympanic membrane. You know this better as your eardrum. And that is right over here. Your tympanic membrane is what separates your external outer ear from your middle ear. This type of membrane, I know we've talked about many different membrane types. This type of membrane is thin, but not too thin, and it can vibrate. The main function of this tympanic membrane is to vibrate. We call it an eardrum, or tympanic means drum, because when you hit a drum, the drum vibrates and creates sound. We're not creating sound with this, but it's vibrating, and that's really important. Sound comes in through our external acoustic meatus, and that sound energy will vibrate our tympanic membrane. Vibration of the tympanic membrane will vibrate the auditory ossicles in the middle ear, which we'll see here in a second. So the main structures I'd like you to know are the auricle, external acoustic meatus with ceruminous glands, and then the tympanic membrane. Those are all part of the outer ear. The middle ear has those auditory ossicles. You've got the malleus first, then the incus, and then most medially is the stapes. Smallest bones in your body. So the tympanic membrane vibrates the auditory ossicles vibrate in succession, and then they will vibrate something called the oval window, which we'll get to in a second. The oval window is this membrane on this organ called the cochlea. But that's in the inner ear. We'll get to that. In the outer ear, This space in the external acoustic meatus is just filled with air. This space here as well is also filled with air. Do you see my pink right here? This is an air-filled tympanic cavity. What's interesting about this tympanic cavity is that it's connected to your throat. It's connected to your pharynx. This connection, this is the auditory tube. You might see it called the pharyngeotympanic tube or the eustachian tube. They mean the same thing. I'm just going to call it auditory tube. This auditory tube, which is lined with elastic cartilage as well as like the auricle. The purpose, why do we connect it to our throat, our pharynx, is because we want to equalize air pressure. If we want the tympanic membrane and the auditory ossicles to vibrate, if we want them to move, they need air pressure to be equalized. Otherwise, if you trap too much air, then there's no room for the bones to move. They won't vibrate. It's why if you have a lot of nasal congestion or if you have an ear infection, your hearing is affected. So far, in terms of hearing, sound waves come in through the external acoustic meatus. We vibrate the tympanic membrane, vibrate the malleus, incus, and stapes. We've gone from the outer ear to the middle ear. The next thing that we vibrate is something called the oval window, and we're getting now to the inner ear. So the oval window is this small membranous window that vibrates because the auditory ossicles vibrate. The oval window is part of this larger snail-shaped organ called the cochlea. And the cochlea is filled with fluid. When the oval window vibrates, it creates fluid waves within the cochlea. Within the cochlea, not only do you have fluid, but you have receptors. The receptors that are not pictured in this diagram because they're way too small, but they're lining all along these spirals. You have hair cells. Hair cells are a type of neuroepithelial cell. They are mechanoreceptors, meaning the stimulus to activate them is some kind of movement. So in this case, it's fluid movement. They detect fluid waves within the cochlea, thanks to the oval window vibrating. When those hair cells get activated, they then send information along these first order bipolar neurons, first order sensory neurons that lead to, the medulla oblongata. This axon of this neuron forms part of cranial nerve 8, the cochlear branch of the vestibular cochlear nerve. I drew a cell right here. You can see the cell body. The cell body is in a cranial ganglion. That's just the beginning of how we hear things. We've, at this point, detected the stimulus, fluid movement, which came from sound, and that then needs to get processed in our brains. Looking more closely at the cochlea, if you can imagine what it looks like in 3 dimensions, this does a decent job of representing it up here. So this cochlea has been cut in half in these places, and then pieces of the spirals have been cut out here, so you look inside of the spiral. What you'll notice, if we just take a look at this one part right here, is that there are 3 different compartments. There's this one here, which I'm tracing in green. There's this smaller one here that I'm tracing in blue. And then there's this large one here that I'm tracing in yellow. 3 different compartments. The one on top, the one that I drew in green, this compartment is called Scala Vestibule. Scala means duct. Vestibule means vestibular. The word vestibular or vestibule, a vestibule is an open space. Pretty vague term for a thing. So yeah, it's an open space duct. Scala vestibule contains fluid, and the fluid that it contains, we call it perilymph. It's basically an interstitial fluid, but it's called perilymph. I'm going to skip down to scala tympani, the one I did in yellow. Scala tympani, the lower compartment, tympanic duct, this also contains perilymph. What that means is, because of this spiral, if I were to unravel this snail shape, if you can see my drawing over here, if I were to unravel the snail shape, so it's kind of like, imagine like a tongue rolled up and then you unfurl that tongue, or like a fruit roll up, you unroll the fruit roll up. The three different compartments, two of them are actually connected. Scala vestibule would be here in green. Scala vestibule is continuous with scala tympani. And then in the middle is scala media. So scala vestibule and tympani are actually one compartment, but because they cross that turn, we separate them into vestibule and tympani. They both contain the same fluid. Scala media, the middle one, has its own fluid that we call endolymph. It's more on the inside, endo, inside. This is also functionally the most important place because this is where you find the hair cells. So we would find hair cells lining this space down here. If you take a look at this histology at the bottom, here is a more zoomed out version of the cochlea. And you can see repeating patterns of those three compartments. 1, 2, 3, 1, 2, 3, 1, 2, 3, 2, 3, 1, 2, 3. These are repeating patterns of those compartments. If we zoom in on one of them, we can see the three different compartments. Here in green is scala vestibule, scala media, I'm drawing in blue, and scala tympani, I'm drawing in yellow. How do we know which one's vestibule and which one's tympani? Media is the smaller one in the middle, but you could easily flip vestibule and tympani. The way that I recognize the difference, I'm going to erase my coloring here. Look for the slant, this sharper slant on one side of media. This sharper slant, that's on the side of scala vestibule. Another way to distinguish, another way to distinguish is to look for more cells on this bottom part of scala media. If we look more zoomed in, this is where we find hair cells, and we're going to take a closer look at that in the next slide. And that would be the side of scala tympani. To help us distinguish between those two sides, we have names for these divisions. This slanted division between vestibule and media, this is called the vestibular membrane. And then this bottom part down here, let me use a different color. This bottom part down here, let me color it in everywhere. This bottom part down here is called the vestibular membrane. Those are what's separating the three different compartments. Let's take a closer look at Scala media, since this is where we actually detect those fluid waves which came from sound waves. Within Scala media, which once again, if we look at this space right here, that's Scala media. Scala media contains a bunch of cells, and collectively those cells are known as the organ of Corti, or you might see it called spiral organ because it's in the spiral. They contain the hair cells, receptors for hearing. Did I call it this? I think I messed up. Let me actually go back 1 second. Let me rephrase these names. This is the vestibular membrane. Did I say this correctly? I can't remember. This is the basilar membrane. It's at the base, the bottom. Basilar, vestibular. Okay. Basilar membrane, vestibular membrane. On the basilar membrane is where you find the organ of Corti. So once again, here is our basilar membrane. Here's the vestibular membrane. Sitting on the basilar membrane is where you find the organ of Corti. About these cells and these structures here. In this organ of Corti, which is zoomed in right here, what we find are a bunch of cells that are supportive, which we're not going to go into detail about. The cells that we care about a lot are these hair cells. And you can see these hair cells have stereocilia. These hair cells, the way they get activated, you have fluid waves in the endolymph. Those fluid waves in the endolymph, they cause vibrations of the tectorial membrane. And they move this membrane called the tectorial membrane. The word tectum means roof. It might also help to think of the word tectonic, if you're familiar with that word. Tectonic, like tectonic plates, are those plates that move during earthquakes, so they can shift. Either way, whether you think of it as a roof or tectonic plates, this is the tectorial membrane covers the stereocell of the hair cells, and they can move in response to these fluid waves. So when the tectorial membrane moves, it shifts the stereocilia. When you shift the stereocilia, you activate hair cells. You activate hair cells, then you activate this bipolar sensory neuron, which leads to the medulla. This cell body, the sensory neuron, is in a ganglion found right next to the organ of Corti. We'll just call it a cranial ganglion to keep it general. So, sound waves come through your sound waves are funneled by your auricle, enter through the external acoustic meatus, vibrate your tympanic membrane, vibrate the malleus, then the incus, then the stapes, vibrate the oval window of the cochlea, cause fluid waves in both the paralymph and endolymph. Endolymph waves cause movement of the tectorial membrane. Tectorial membrane shifts stereocilia, which activates the hair cell, which activates that first order sensory neuron, which goes to the medulla, and then we'll see it go to the thalamus and then to the auditory cortex and the temporal lobe. That's how you hear things in a nutshell. Like I just said, sound waves come in. That's step one. Vibrate the tympanic membrane, then the malleus, then the incus, then the stapes, then the oval window. You get vibrations in the endolymph, and that's what causes stereocilia of hair cells to bend. If you want fluid waves or any waves to continue, you need an outlet for that energy. Like if you imagine punching one of those punching bags, those speed bags. If it's just hanging and you punch it, no problem, you punch it and it'll move back and forth. But if there's a wall, if it's up against a wall and you try punching it, the bag's not going to move because it's being supported by the wall. If you want the bag to move, it needs space. It needs an outlet for that energy. Similarly, if we have energy waves, fluid waves coming through the cochlea, there needs to be an exit for these waves. I'm not saying that the fluid has to leave, but the energy of that movement has to leave. So we go from movement of paralymph in Scala Vestibule, it turns that corner, and it comes out Scala Tympani, and that energy gets vibrated out another membrane called the round window. Confusing, there's an oval window and a round window. There's a difference. The oval window is the one in contact with the stapes. The round window is on the other side with Scala Tympani, and it's just for letting that energy exit. That allows energy to dissipate. So that's the function of the paralymph, to let the energy pass through. The endolymph is to move the tectorial membrane. Paralymph is to let the energy pass through. That's, again, in a nutshell, how we hear things. When we have a sense of balance, our vestibular sense, head movement, we rely on a different organ that's next door and actually physically connected to the cochlea. It's called the vestibular complex. You might see it called vestibular complex, vestibular apparatus, it's the same thing. Vestibule, again, just means an open space. So a very generic term for this type of structure. But once again, what this helps us sense, it senses balance, specifically head movement. Our head movement helps us balance. Take a look at this drawing of it right here. This is the cochlea. Roughly this part, while the vestibular complex includes a bulge here, and then 3 loops. 2 bulges, 3 loops. In these bulges and loops, what we have is fluid. So this is fluid filled as well, just like the cochlea. It's the same endolymph that's in the cochlea. It's continuous. It's the same endolymph that's in the cochlea. Fluid movement, endolymph movement, stimulates hair cells. And stimulation of these hair cells isn't for hearing, it's for sensing head movement. So it's really weird and interesting that we can use the same exact receptor type for a different, completely different sense. One is detecting movement of sound waves, one is detecting head movement. And here you can see an example of this in this picture. Let me use a different color. In this picture, we have hair cells. And if there's movement, then there's a bending of the stereocilia. Head movement can bend these hair cells, the stereocilia of the hair cells. let's dive deeper into what are these 3 loops and what are these 2 bulges. The loops are called semicircular canals. They're half circles, semicircular, and canals, they're thin passageways filled with fluid. The reason why they're circular is so that you can detect rotational acceleration. Detecting that you're spinning or your head is turning, whether it's left, right, side, side, back, forth, those are the 3 basic head movements that we can do. I know there are special terms for them, but I don't remember them off the top of my head. One's like pitch, one's yaw, something like that. But anyways, that's why we have 3 different semicircular canals, and for those 3 different planes. If I zoom in on one of these loops, what we're seeing something that looks kind of like this. Filled within, you've got your endolymph. So in blue here, I'm drawing endolymph. And then you've got this bulged region called an ampulla. This bulged region is called an ampulla. Ampulla is a general term for a bulged region. This bulged region is important because it contains the hair cells. So let me draw some hair cells. Attempt to. I'll just go draw one for simplicity. Definitely not to scale. There's a hair cell. This hair cell is contained within a gelatinous material. So let me attempt to draw a gelatinous substance surrounding the hair cell. Whenever there's fluid movement, so let's say you turn your head and the fluid, the endolymph, moves through, that'll cause bending of the stereocilia. And when those stereocilia bend relative to endolymph movement, then we send information through a sensory neuron, a bipolar sensory neuron, to the medulla oblongata, so that we can further process our head movement. And this axon would be part of the vestibular branch of cranial nerve 8. That's how we detect head rotation, rotational acceleration, whether it's left, right, back, forth, side, side. Any kind of rotation, it's because of a semicircular canal. Hair cells being activated. Those are the 3 loops. The 2 bulges are shown here, whoops. And here. The bulge on top is the utricle. The bulge on bottom that I'm doing in green or blue, excuse me, is the saccule. The way I remember this, the saccule, if you're holding a sack, sacks hang down, sacks on bottom. Both of these are for linear acceleration, not rotational, but linear. So if you're going in a car, for instance, if you're driving in a car and you go from 0 to 100 real quick, that would be in the horizontal plane, that would be activating hair cells in the utricle. For the saccule, it's in the vertical plane. So for instance, if you detect falling down from in an elevator, you get that sudden movement, that drop, and you feel that weightless feeling, that's detected by hair cells in the saccule. There's a slight difference here in how there are hair cells, there's a slight difference in how these hair cells are activated. These hair cells are also embedded in a gelatinous material, but on top, or actually within this gelatinous material, you have these things called otoliths. They're represented here in this very light blue stuff. And you can hopefully, I know it's small, but you can see hair cells all around here. Otoliths sit on top of the hair cells. And let's say these fingers are hair cells. If you have something heavy sitting on top of these hair cells, let's say you move quickly this way. If there's nothing sitting on top of the hair cells, nothing happens to the stereocilia. But if something heavy sits on top and I quickly move, my fingers bend, the stereocilia bend. The otoliths are critical for providing enough weight so that when you move quickly, the stereocilia will bend. That's how you detect linear acceleration. Otolith, oto means ear, lith means rock or stone or something mineral. So literally means ear stones. You have little chalky stones in your ear. I think it'll be useful if we see some histology of this, of the hair cells and a few other receptors. So I'm going to switch over to histology and also a model to show this in a bit more detail. Let me show this ear model first, and then we'll move on to histology. So this is in the Autonomic Nervous System and Senses video page, or you can search for this on YouTube, ear models, Cochlea, vestibular. What we have here are models of the ear. So we're going to look at this one. We're looking at the left ear, pointing at the auricle. Just label everything. Auricle, external acoustic meatus, tympanic membrane, that's all part of the outer ear. See the passageway here? That's our auditory tube. It's a little hard to see each individual one, so I'm just going to group them together. These are the auditory ossicles. And then over here, you can see the loops of the semicircular canals, and then a bulge, and then over here it's a cut cochlea. So really this is the vestibular organ and the cochlea. If we look at a closer up model of the vestibular organ and cochlea, here is a larger model to show all the different parts. So this snail-shaped thing, that's the cochlea. The one on this side, this is the oval window. You can see the semicircular canals here. You can barely see the utricle and saccule. You see one bulge here and another bulge here. Utricle, saccule. We'll be able to see it better when we spin it around. Now you can see them pretty well here. Utricle is on top. Saccule is a little bit lower down. There's that oval window. Looking more at the cochlea, look at how it spirals around and around and around. If we cut into the cochlea and look inside, we'll be able to see the different ducts. So let's remove that half that's done here in this model. How do I know which duct is which? Let's look at this grouping of three. We see a slant right here. The more slanted one is the vestibular membrane. Which means this other slant is the basilar membrane. That means here in the middle, you can see the organ of Corti a little bit. And you can see as a whole the scala media. This side of the basilar membrane would be scala tympani. This side of the membrane, of the vestibular membrane, would be scala vestibule. You can also see the two branches of cranial nerve 8. Here we have the cochlear branch. And here, different color. Here we have the vestibular branch of cranial nerve 8. The cranial ganglia where we find the cell bodies of bipolar neurons for hearing, the first order sensory neurons for hearing, they're found here in this cranial ganglion. Here's another model that shows not only the cochlea cut how we just saw it, but also cut the other way longitudinally. So you can see the three different ducts. We've got media. I can't tell from here, but one of them is vestibuli, one of them is tympani. Spirals all the way around. This model shows a blown up cross section of the cochlea. We have scala vestibuli, scala media, scala tympani. You've got your vestibular membrane, your basilar membrane, tectorial membrane, represented here are hair cells with stereocilia within the organ of Corti. And then a sensory neuron will get activated. It's bipolar. You can see a ganglion right here. And these axons will continue on as cranial nerve 8 to the medulla. Back to that blown up model of the cochlea and vestibular organ. This is the oval window. The oval window is larger and more oval. This is what's directly in contact with the stapes, the most medial of the auditory ossicles. We spin this around on the posterior side of this organ. It's rounder and smaller. This is the round window. Lets energy dissipate out. It'll be useful to see the histology of hair cells. It looks a lot like what we saw in the models, kind of, but I'll show you again. Here you can see at 40x, a whole cochlea cross-section. You can see a grouping of 3, 3, 3, all going all the way around. If we zoom in, this is at 100x. Here's a grouping of 3. You can see it's lighter. You can see a slight division going this way, and then a darker division going this way with more cells. So that means this is Scala Vestibule. This is media. This is tympany. This is at 100x right now, the cochlea. If we go to 400x, here we're zooming in on the organ of Corti. It's kind of hidden, but what we can see here, this extension, it's a little bit curled up, and that's just an artifact of, because when we cut this, not everything's going to stay fully intact. But this thing is the tectorial membrane. Down here is the basilar membrane. And these cells, these are the hair cells. The stereocilia are really small, so you might just see a little bit of fuzziness. But those are the hair cells, and the tectorial membrane will be sitting on top of it. While we're here, actually, let me show you one other thing in this video. Here you can see Scala Vestibule, Scala media, Scala tympani. Hair cells would be right here. The neurons that communicate with these hair cells would extend this way. And so this is the nerve. If you look closely in this region right here, there's a lot of dots. These are neuron cell bodies. This is the ganglion. And if we go closer, this is 100x. You can see the hair cells right here. And then if we move over to the right, now we see neuron cell bodies of the ganglion. of this cranial ganglion. For bipolar neurons. That's histology of hair cells in the cochlea. We don't have the histology of hair cells in the vestibular organ, but the hair cells would look similar. Let's compare this to other receptors. Hair cells are specialized receptor cells, neuroepithelium. When we look at gustatory cells of the tongue for taste, these are also specialized cells, not hair cells, they're gustatory cells. You can see the surface of the tongue right here, 40x, we don't see a lot. Let me go back a little bit. This is 100x. You can see that there's this very deep wavy texture to the surface of the tongue. If you recall, we have bumps on the tongue. Those are called lingual papillae. By having texture on the tongue, it's good for gripping and more surface area for having receptors. So we're going to look closer at these lingual papillae to find the taste cells, the gustatory taste cells. So we go to 400x. If you see this bunch of cells right here, it's a bit lighter. There's another grouping right here. What we're seeing here, if I blow it up, what we're seeing here are a bunch of cells smushed together in a grouping like this. The surface of our tongue would be here. And if you eat something or taste something, those molecules make their way to this taste pore and get detected by the taste cells, these gustatory cells. When these gustatory cells are activated, they activate a bipolar sensory neuron that goes to the brain. This axon will be part of one of three potential nerves, depending where on our tongue we're tasting. If you recall, for taste, we could use cranial nerve 7, facial, cranial nerve 9, glossopharyngeal, if we're in the latter third, or in the very, very back, even in our pharynx, cranial nerve 10, vagus. So gustatory taste cells are also a specialized receptor cell. For smell, we don't actually use special receptor cells. We use free nerve endings. They're bipolar neurons because this is a special sense, but we use we use free nerve endings. So we can see a free nerve ending coming up here. All this texture you see here at 40x are folds and space within our nose to help us breathe. So this lumen here, that's the nasal cavity. So we're going to zoom in on, oh, and you can also see hyaline cartilage. This dark purple is hyaline cartilage. It's hard to see right now. Let's go closer. This is 100x. You can start to see the hyaline cartilage better. And you can see the epithelium on either side facing the nasal cavity. Again, this is at 100x. At 400x, what we're looking for, it's a very, very slight thing. We're looking for some light colored cells that are a bit long. So they're going to be in this region towards the surface, because if you want to smell something, you got to have your dendrites right against your nasal epithelium. Hopefully it shows up for you, but for me, I can see one right here. The cell is a little bit longer. If you can imagine, its dendrites will stick out this way and its axon is going to come out this way. Let me redraw off to the side. Dendrites, longish cell body, then axon would come out. That is an olfactory sensory neuron. Olfactory sensory neurons are free nerve endings, and they're bipolar. You can see another one right here. I think I've covered another one up, but you can find another one right here. It's the longer cells. The rest of the cells are supporting cells, which we're not going to go into much detail about. So we've seen specialized receptor cells of the tongue, gustatory taste cells, specialized receptor cells of the ear, hair cells, free nerve endings, olfactory sensory neurons. In the next video, we'll take a look at the retina, where we'll see photoreceptors, another type of specialized receptor cell. And I just want to remind you of complex neural receptors that we've already seen. We've seen Meissner's corpuscles and Obsidian corpuscles, the histology of these. Meissner's corpuscles are in the dermal papillae of the dermis. So here we are in skin. This is thick skin. You can see the thickness of the stratum corneum. Here we are at 400x. This structure here this is a Meissner's corpuscle. Which is a complex neural receptor. If you can imagine, it's carving up dendrites, and the axon exits out. Its cell body would be often some ganglion, whether it's a cranial ganglion for the face or a dorsal ganglion for neck down. Yeah, so it's connective tissue covering up a pseudounipolar, dendrites of a pseudounipolar neuron. Similarly, for a Obsidian corpuscle, these are much larger. Previous one was at 400x, this is at 100x. Obsidian corpuscles are also complex neural receptors. Covering up dendrites of a pseudounipolar neuron. All right, that was a quick recap of different structures of sensory receptors, free nerve endings, complex neural receptors, specialized receptor cells, and also an overview of hair cells and how we hear things and how we detect head movement. In the next video, we'll talk about the eye, special structures of the eye, and how photoreceptors detect light and send it to a pathway to help us see things. So I'll see you in the next video. Please let me know if you have any questions. Leave it in the comments, in the discussion. I'll see you in the next video. Talk to you later.